Anybody got struck by lightning or flooded out yesterday? It's pretty soupy last 2 days. Okay, so we are now talking about neuroglia, and glia actually means glue. So these are the cells that hold the nervous system together, physically providing them support, but also helping with nutritional transport to and from neurons. Not everything gets to neurons in every single part of the brain. So some of these neuroglial cells will determine whether or not something that's in the bloodstream is going to get to a neuron or not. They'll also interact with ependymal cells and determine what's getting from the blood to the ependymal cells and then into the cerebral spinal fluid. So these cells will be doing that. They'll also help to repair any central nervous system damage. And then there are also some stem cells in there. Finding out a lot more about the nervous system stem cells. In fact, this weekend I was watching my favorite program, The Medical Mysteries, and there was a young girl who started drinking a lot of water. And they tested her for diabetes mellitus, and it wasn't diabetes mellitus. What should they have then checked her for? What's the other kind of diabetes? Well, there's the adrenal, but what else? Drinking a lot of water, but no sugar coming out in the urine? Insipidus. There were 4 nameless doctors, again, 4 idiots who didn't pay attention in 301. Turned out it wasn't damage to the posterior pituitary, but after a long time not figuring out what was wrong with her, all of a sudden her eyes started going kooky. And then they said, ooh, they got really scared. Their eyes are going kooky. And so they did an MRI and they found a brain tumor. They could have figured this out a lot earlier had they looked at the hypothalamus and the posterior pituitary. Anyways, the diabetes insipidus is a problem that is going to be posterior pituitary problem, but also what was turned out to be the cause of that brain tumor were stem cells. Stem cell brain tumor. That's very, very rare, highly unlikely. Usually it's some other kind of cell. Gliomas, they're common, but stem cell brain tumor is very uncommon. Okay, so types of glial cells. Astrocytes, named because they have all these cell processes that look like rays of stars going out in different directions. Small little phagocytic cells, microglial cells. They go around phagocytosing damaged cells in that area. The oligodendrocytes, these cells put out several processes, foot processes or hand-like processes, and they wrap sections of the central nervous system axons, creating myelin. Similar cells will do that wrapping in the peripheral nervous system. But in the central nervous system, where space is of an essence, you don't want to have a lot of cells taking up space. So you have one cell doing the job of several cells that you could have in the peripheral nervous system. So this oligodendrocyte will wrap this part of this neuron here, the middle neuron, and then also this neuron to the left. Here's one that is wrapping the next segments of both neurons. Here's one, you see it wrapping part of one neuron and then another part with astrocyte foot processes in the middle. This is not cut away. It isn't wrapping multiple times. It's just going around that axonal process once. So this would be an unmyelinated cell process. So astrocytes, their job is to support unmyelinated axons. They also have foot processes that cover majority of the blood vessels in the central nervous system. And they then determine, based on what they endocytose, what will get to neurons, and then also what will get to ependymal cells. So they'll be forming part of the BBB, blood brain barrier. There's BBB, not there in all areas of the brain, because in some areas you have to get large things in and out of capillaries. So in the hypothalamus, blood-brain barrier is missing, because you have your releasing hormones have to get in, and you have to have access to the feedback hormones to those hypothalamic neurons. And you have to have in the anterior pituitary, the hormones getting out to stimulate the anterior pituitary, and the hormones that are being released by the anterior pituitary having access to the blood. So those areas also, no blood-brain barrier. Posterior pituitary, ADH and oxytocin have to have access to the blood, so no blood-brain barrier. The brain stems, you have to monitor the blood for pathogens. And so there are areas that are linked to your vomiting center where you don't have a blood-brain barrier, so that pathogens will be recognized by those areas of the brain and then trigger vomiting. So you clear out a potential source of pathogens and just clear your stomach out. And that probably is a good way, good route for bacteria coming in. So again, linked to the vomiting center. Upstairs, there's Frederic, I don't know. He's from France, Messier. He's working on stem cells and very involved in the cutting cells and then stimulating stem cells and having the central nervous system reform these damaged neurons. It was once thought that central nervous system could not regenerate. Once it was damaged forever, but turns out, nope, there are stem cells here, and cells can regenerate in the central nervous system. Out in the peripheral nervous system, the cells that are acting like glial cells, supporting the nerve cell bodies and blood vessels in those areas, these are referred to as satellite cells. They will support the cell bodies in the ganglia, which are collections of cell bodies outside the central nervous system. So all these pink cells, these are neuronal cell bodies. Perikaryon, peri meaning around, karyon meaning nucleus. This is going to be full of mitochondria and rough endoplasmic reticulum, Golgi apparatus. They just haven't drawn it in there. But if you come back and you look at this cell body, it shows you rough endoplasmic reticulum, mitochondria. They haven't drawn the Golgi in there, but all the organelles of the cell body will be in this pink amorphous stuff that the artist has drawn there. Here, where you see the light micrograph, you see the grainy texture to this cytoplasm. And so that's indicating there are organelles in that area. The satellite cells, they also are controlling substances getting to and from the cell body, so waste product, nutrient exchange, probably some gas exchange control as well has been hypothesized as to function of these small little satellite cells. All these small little nuclei there. Those are the nuclei of the satellite cells. The neuron in the peripheral nervous system When it's myelinated, it's going to be myelinated by Schwann cells, capital SCHWAN, and this is a eponym. It hasn't been gotten rid of as far as I know. They're still called Schwann cells. They wrap multiple times one section of one axon. So axonal ratio is one Schwann cell to one axon. In the central nervous system with oligodendrocyte, it's one oligodendrocyte to 2, 3, a few segments of axons. So here it's a one-to-one relationship. Multiple Schwann cells wrapping each little segment of the axon, and then the nodes of Rambier in between. There also are a second population of Schwann cells, or a different differentiation factor where the Schwann cells will also support a multitude of axons, and they do so by just holding them in place, not wrapping them multiple times, but just going around each axonal segment once and just supporting it in position. So all of these axons will be considered unmyelinated axons. And you see them in the EM here. You have the axon wrapped with one layer of Schwann cell plasma membrane, as opposed to on the left, multiple layers of the Schwann cell plasma membrane. The perikaryon, anything that's around the nucleus. So rough endoplasmic reticulum, mitochondria, Golgi apparatus. The term Nissl body or Nissl substance, another eponym, NISSL is an eponym. That's referring specifically to rough endoplasmic reticulum. And the reason they make a big deal about this is if you can find a process that leaves the cell body that contains no rough endoplasmic reticulum, you can then say that that is the axon. The rough endoplasmic reticulum will extend into the dendrites, but it will not extend into the axon. And the axon, all the way down to its telodendria, will be devoid of that rough endoplasmic reticulum. So it's really hard to find that process when you're sectioning through the brain, or the peripheral nervous system. But if you can find that one process that has no rough endoplasmic reticulum in it, then you can say that is the axon of the neuron. We talked about the axon hillock already, talked about the initial segment and the axon, the telodendria, and also the dendrites, so we won't be going over that again. But here you look at the light micrograph. The rough endoplasmic reticulum goes out into all these processes, but it stops short of that process. So this is the axon. I always wonder how many slides did they have to go through to find that particular axonal process. It's really hard. When you try in lab, it's really hard to find those. So now we're going to be talking more about postsynaptic cells, presynaptic membranes, and how communication will happen within the central and peripheral nervous system. The types of neurons, if the cell has no particular designated axon, it's referred to as an anaxonal neuron. So what that means is any of those processes can function as a dendrite, can also function as an axon. So these clever little cells can send information in any direction they want to. They will just select that process in that direction that they want to send information to be the axon. and the others will then be receiving information. So if they want to send information off to the upper right corner of your brain, this will be the axon. But if they want to send it to the lower part of the nervous system, then this will be the axon. If you want to send it to the left, then that will be the axon. Other information, other processes will serve to collect information from other parts of the brain, and then depending on what direction the neuron wants to send information, it will select the appropriate process to act as the axon for that particular integration and then sending of the information. All the other neuron types, they're limited in that they can only collect information via one or several processes, and they always have a dedicated axon. That axon cannot bring information in, it can only send information out. We have things that are specifically, someone was coming from here. It's like, what? I never heard my cell phone make that noise. Anyway, okay, so in special senses, we're going to see a lot of bipolar neurons. So in the retina, in your olfactory system, in your in the special senses, you're going to see bipolar neurons. In the somatosensory system, you're going to see these pseudo-unipolar neurons. They're referred to as pseudo-unipolar because it appeared when they first looked at these neurons that there was just one pole, that the information seemed to come in and out of one area of the cell body. And so they earlier anatomists called them unipolar neurons. But then when they got the light micrograph, when they got the electron micrograph, they were able to magnify that one pole, and they saw that there were two processes, but they were wound around each other really tightly. So on low magnification, it looked like one process, but on high magnification, they could see there are two processes. So they changed the name to pseudo. It appears unipolar, but it's really not unipolar. One of these ends will be linked to a long process that is dedicated to bringing information to the cell body. So these sprouts here are referred to as dendrites. And then this process is going to take information away from the cell body, and these will be your synaptic terminals or telodendria. Some texts refer to this as a dendrite and this as an axon, because this process brings information to a cell body and this one takes information away from the cell body. But this Martini, he's calling both of these axons. I myself prefer this process to be described as a dendritic process, bringing information towards the cell body, and keep dendrites as afferent information coming in, and the axon as efferent information leaving the cell body. This neuron here is called a multipolar neuron. When you have more than two poles, then you go from bipolar to multipolar. Most multipolar have a lot of processes coming into, a lot of dendrites coming into the cell body, and then a dedicated axon leaving the cell body. So all your motor neurons that we'll be talking about in the next chapter, they're all these multipolar neurons. Interneurons, all multipolar. They're going to be involved in integration, so take information from a lot of different areas and then send information to the appropriate part of the central nervous system or peripheral nervous system. all your association neurons. We haven't gotten to identify what an association neuron is yet, but you're supposed to come into this course with that general knowledge. Association is communication between one part of the central nervous system and another part of the central nervous system. So frontal and parietal lobe, parietal occipital lobe, occipital and frontal lobe, right hemisphere, left hemisphere, your cerebellum and your basal nuclei, all of these intercommunication between one part of the central nervous system or one segment of the spinal cord with another, those will have be done by association and interneurons. The classification, is it a receptor? It's probably going to be located in the periphery, so you can monitor your external environment if it's hooked up with the somatic nervous system. But if it's in your organ systems, there will also be receptors there to monitor your organ function. So interoreceptors, those are inside your body. Exteroreceptors, interoreceptors will monitor what's going on inside your body. Exteroreceptors will monitor your interface with your external environment. And proprioceptors will tell you about position of your muscles and your joints and whether they're being stretched or not stretched. Interoreceptors are going to send information into the central nervous system from areas like your digestive system, your respiratory system, cardiovascular system, your urinary system, reproductive system. And then you'll have integration happening and then a response going back to those internal organs. So your autonomic nervous system, the preganglionic and postganglionic neurons will go back to the various organ systems that interoreceptors were monitoring. The exteroreceptors telling you about your interface with your environment, they'll send information about touch, temperature, pain, vibration, stretch of your muscles, stretch of your joints, and they'll then have integration happening, and then somatic neurons will respond, and you'll have your skeletal muscle react based on the information here. If you touch something hot and you pull your hand away, you have something hot and or cold, your muscles may start to shiver. So it depends on the integration that's happening in the central nervous system, what your skeletal muscle will do with that information. Special senses, sight, smell, the hearing, that also will be monitored by exteroreceptors. So photons of light, the chemicals on your tongue, the vibrations from your environment going in, you'll have special sensory receptors for vision, for smell, for taste, for proprioception, etc., in these special organs that we'll be covering later. And those can also elicit certain motor responses. So if there's loud noise, you look up, where's that noise coming from? Is it dangerous? No, I can go back to writing my notes. And so you have these wired responses to the noise. Types of synapses. We have synapses between neurons. So the presynaptic, postsynaptic neuron interface. The axon here is binding to the cell body, and if you look closely, it's also binding to the dendrites. So we have axodendritic. We have axosomatic. to the cell body, so axosomatic, axodendritic. There are also these little collateral branches. They could be going to a neighboring axon and forming an axo-axonic. Synapse. So three basic types of synapses for neurons. Interacting with the axon of another neuron, with the dendrites of another neuron, or the soma of another neuron. Neuromuscular junctions, we've covered that already. Got a motor neuron and the muscle cells that it innervates, forming a motor union. We've also got motor glandular function. We loosely talked about that in the endocrine system, that we have the nervous system capable of stimulating an insulin released from the pancreas. Also, the release of norepinephrine and epinephrine from the adrenal medulla from modified postsynaptic neurons. Also, in the pancreas, the activation of beta cell, that would be neuron to gland cell interaction. So neuroglandular is another type of synapse. Here we've got a cell body with a lot of dendrites. It's all been cleaned off. Over to the side here, we've got that same cell body with all the presynaptic input to that cell body. So all these little blue buttons, those are other neurons that are synapsing with the dendrites, with the soma and also with the initial segment of that axon. So you have all these inputs. You have no idea what the input is. It could be an excitatory input, it could be an inhibitory input, but the cell body's job is integrate all that and determine if there is enough depolarizing information then to initiate action potential in the axon. Action potential goes down to the telodendria. The arrival of the action potential there opens calcium channels. Calcium comes in, interacts with the filaments of the vesicle, get the vesicle pulled to the plasma membrane, neurotransmitter released into the synaptic cleft. That space between pre and post synaptic neuron membrane. So in the light micrograph, they've colored it so the vesicles are all green, the mitochondria blue, cytoplasm the pink. The synaptic cleft is the kind of a bluish color, postsynaptic membrane, more cytoplasm, so predominantly pink again. So some of these processes may also be glial cells that are supporting the position of that neuron, and they also could be the axons of other neurons interacting with that cell body and dendrites. The type of input and output, it's not clear what's happening here, but multiple inputs from multiple neurons. You have one axon going out and it's branching to synapse with 3 visible postsynaptic neurons here. Don't know what this is going to. You can see one synapse, but here we've got at least the recruitment of 3 more downstream neurons. The way the nervous system is organized is that we have different types of sequencing for these neuron-neuron interactions. If we want to recruit widespread response, we want a sympathetic nervous system recruitment of everything, vision, muscle, heart, all at once to mount this fight or flight response. We use a divergent pathway where one upstream neuron may recruit 32 or more downstream neurons. So we have one branching here, we have another branching here. We don't know what these neurons are going to do, probably branch again and recruit more, up to 32 downstream neurons recruited by one upstream. So the fact that 1 goes to 2 and each one of these goes to 3, we have a divergent pathway. If we want one organ or one part of one organ to respond, like parasympathetic nervous system, you want to have very discrete, very localized response. You have a convergent pathway where several presynaptic will converge to one postsynaptic, or you have serial processing. That also could be one-to-one relationship. The parallel processing, the difference between parallel processing and divergent is that after the initial divergence, it's all serial processing from that point on. So we don't call that serial processing when it diverges first. We usually be a little more specific than parallel processing, but because that could imply that you had a bunch of serial processing going on, just lateral to each other. So in most textbooks, they'll say parallel after, or parallel after processing, to talk about the pathways after the initial processing and divergence. You have a parallel circuit. The reverberating circuit can be to stimulate the first neuron. So the second neuron may come back and re-stimulate the first neuron, so you get positive feedback, stronger and stronger link between the first and the second neuron. Or the reverberating circuit may be inhibitory, where the second neuron turns off the first, which is more common in the body. That once you get something initiated, the cell downstream then turns off the upstream stimulation. So reverberating circuits, parallel, serial processing, convergent, divergent, depending on what you want to do with your central nervous system, you'll have a different type of circuit organization. You want to recruit widespread, have it diverge a whole way. If you want to localize the response, have a converging system. You want very, very specific from one area to one area, then a serial processing would probably be the type of circuit that you want. If you want something to increase the response repetitively, get stronger, 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 you have positive feedback reverberating circuit. If you want it to turn off with the second cell stimulation, then negative feedback reverberating circuit. So the last slide here kind of summarizes everything that we've talked about. The peripheral nervous system, if you have a collection of cell bodies outside the central nervous system, you call that collection a ganglion if it's single, or ganglia if it's plural. Inside the central nervous system, collections of cell bodies we'll talk about as cortex, but if they're clumped together, we'll call them nuclei. So collections of cell bodies within the central nervous system that aren't spread out on the surface, you'll call that a nucleus or a nuclei. The pathways can be motor output, and that will be continuing on into the peripheral nervous system. The white matter in the central nervous system will connect with myelinated nerve at some point in the peripheral nervous system, and it might change to unmyelinated at some point in the peripheral nervous system. The white matter that's in the peripheral nervous system may have unmyelinated neurons in that bundle. So a nerve is going to be a collection of myelinated processes, but if you look really closely, there are unmyelinated neurons there as well. But that myelination gives this white appearance to the neuron. The neuron is either going to be afferent or efferent. When you bundle it together to form a nerve, you bundle efferent and afferent together. So a nerve is not a neuron. I know that my TAs have been emphasizing this because they're really frustrated because students aren't getting it. That a nerve and a neuron are not terms you can use interchangeably. A neuron is a cell body and processes. A nerve is a collection of processes, can be afferent and efferent together. So multiple axons and dendrites bundled together. Collection of cell bodies in the central nervous system. So here, 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 those are nuclei. The white fiber tracks in the central, the collection of myelinated and unmyelinated in the central nervous system are going to be referred to as tracts. And when the tracts are stuck together around in between nuclei or around the gray matter in the cord, then they can be referred to as columns, so chunks of this white matter. Higher centers, they're referred to as above the brainstem. So the brainstem and the spinal cord are older parts of the nervous system. So when we talk about humans and the higher centers, we're talking about things that are up here in the telencephalon, in the new cortex. And the brainstem and spinal cord more as the lower parts, or the older parts of the nervous system. The higher centers, that's where we do the most complex integration of sensory information. Brainstem and spinal cord, those don't process they don't do the conscious level processing, which humans tend to egotistically think is so wonderful. The brainstem and the spinal cord, they will respond more with a reflex type of response to sensory input. So humans tend to think that is more of a baser type of function. The centers will have centers that handle sensory information, like the postcentral gyrus. Centers that handle motor output, the precentral gyrus. Centers that handle visual processing, auditory processing. So specific processing functions, those will be referred to as centers. The arrow tells you it's headed out for processing, so that's ascending. That will be afferent information or sensory information. The gray, the arrow, find it down here going somewhere, that's efferent information or motor information. So spinal cord, you cut the cord at the cervical level or at the end of the cord around the top of the lumbar end of the thoracic. That's where the gray matter is the largest. That's where you have the most neurons that are involved with your upper extremities and your lower extremities. So most gray matter in these areas because you have motor nerves going out to the upper extremity. You got motor neurons going out to the lower extremities. We also got sensory input coming from the lower extremities and sensory input coming from the upper extremities. So this gray matter is going to be enlarged at the cervical and the lumbar regions. The sacral cord, less going out and coming in at that level. Sacral nerves, some pelvic floor muscles, autonomic, parasympathetic output. Thoracic, most of the organs in your thoracic and abdominal area, they're run by the vagus nerve. The vagus nerve accounts for around 90% of your autonomic parasympathetic output. So what's here in the thoracic area is controlling visceral organs. What does show up here is an enlargement in the lateral area. It's the lateral horn of the gray matter, and that's specifically for visceral motor and visceral sensory. But the motor, the primary somatic motor and somatosensory, very small in that area because most of the work is done by your autonomic nervous system, which is either in the lateral cord or it's coming from your vagus nerve, which is exiting your cranial cavity. Conus medullaris is this tapered end of the spinal cord. Just comes to a point, so conical cone shape, conus medullaris. The nerves that are continuing down are referred to as the cauda equina, the horse's tail. Those neurons and nerves were, the bone formed around them when the cord actually was down to the very tip of the vertebral canal. The body accelerated in growth, the cord did not keep up. So the cord receded, the nerves elongated, and you have the horse's tail left over. The dorsal root ganglion is a good landmark to know. That tells you you're on the dorsal side of the spinal cord. You can also look at the center here. On the dorsal side, there is a sulcus, which really means that if you wanted to get in there and pry the two sides apart, you'd see that they could be, that the pia mater could be severed and the two sides could come apart. But on the anterior side, on the ventral side, there's no need to do that. There's actually a space there. There's a fissure there. It's referred to as the anterior median fissure. So if you look at the cord, there are several ways to tell anterior from posterior. If the dorsal roots are there, use those. They're not going to be any ventral roots. And you can tell that without actually having to cut the cord. The other things you can do superficially is look for the sulcus, that will be dorsal. Look for the fissure, that will be anterior. When you cut the cord, then look at the gray matter. The blunt, round, anterior horn is always be rounder and bigger than the dorsal horn. Dorsal horn is connected to where the dorsal root axons are coming in. Those will be cell bodies that are related to sensory information that's coming in. And then you need to distribute it throughout different pathways. So cell bodies for relay of information might be located in these posterior horns here, all the way down. And the ventral horn usually is blunter and rounder than the dorsal horn. It's more tapered. It also goes all the way to the surface. The gray matter goes all the way to the surface of the cord, whereas the ventral roots tend to be separated by white matter from the surface of the cord. When you look at the cord and dissect off the dura mater, you'll see pia mater attached right to the surface. It's attached to the surface of the brain, it's attached to the surface of your spinal cord. Cut that, and then you see the actual cord. The fissure, there'll be a space that you can see there between the right and left sides of the cord. This denticulate nucleus that's labeled here, I had to go and look this up for myself. Why are they saying that looks like a tooth? I don't see it. Well, it looks like the teeth on a serrated saw or a serrated knife. When you look at the saw, they call those jagged edges of the saw a serration, and that also, or notched appearance. When you look at these ligaments where they're going out through, getting pulled out and attached to the dura mater, it gives it a notched or serrated appearance. The arachnoid is going to be connected to the pia by these delicate little filaments, and that is what gives the arachnoid its name, the spiderweb appearance. That's where your cerebrospinal fluid will be bathing the surface of the cord, and then this white, really shiny substance that disappears as the nerves disappear, that's the dura mater. The other place cerebrospinal fluid is located is in the central canal of the spinal cord. But usually you can't even find it in a cadaver because everything has gotten so much water has come out of the tissue that the central canal is just kind of all squished down on top of itself. So here the artist and people have done this, they've actually dissected away. Got the dura mater that is interfacing with the bone, and there'll be some fat there for protection. So dura mater will actually become the covering of the nerves as they exit the vertebral canal. That will be connected to the arachnoid, and then the arachnoid will be connected to the pia, which is on the surface of brain and spinal cord. This space here, the subarachnoid space, is where cerebrospinal fluid will be, and also in the central canal of the spinal cord. So simplest way, if you can see the dorsal root ganglion, is look for it. It's covered with the dura mater, so you can't tell is the dorsal root associated with that root, or is the dorsal root associated with that root? So then you look at the cord itself. If you find a fissure, then you know this is anterior. If you find a sulcus, then you know that's posterior. The posterior is dorsal. The dorsal root is associated with the dorsal root of the nerves. Ventral root is going to be motor. This is information leaving the spinal cord. Dorsal root is sensory information coming into the spinal cord. These other little bulges here, these are autonomic sympathetic ganglion, and they're going to form a chain that will continue up and down right next to the vertebral bodies. There'll be a chain of ganglia that will go all the way back up to the cervical region and all the way down to the sacral region. If you can draw an arbitrary dotted line through the center of the central canal of the spinal cord. I tried really hard to get that line in the center of the central canal, and I could not do it. So just pretend it happens to actually go right between the visceral, sensory, and visceral motor nuclei. So that was pretty cool. But I couldn't get it in between, right centered in that central canal of the spinal cord. But that arbitrary dotted line, if you can make that in your head, superimpose it on the spinal cord, for the gray matter, it's really cool because everything posterior is going to be sensory. All that gray matter in the gray cord, that's all going to be sensory gray matter. Everything that's anterior, ventral to that, that's all going to be motor. So here in the lateral horn of the spinal cord, that shows up in the thoracic area and isn't in the lumbar area, isn't in the cervical area. This is all related to your viscera. This is autonomic nervous system. Visceral sensory, the cell bodies here that are going to be taking information up to your brain, telling your brain about, oh, we've got pain in your heart, pain in your gut, pain in your liver, maybe getting it right, probably not getting it right. That is where the cell bodies are going to be located. So sensory information from your organs will come in the dorsal root and synapse in the lateral horn, and then go to processing centers up either in the brainstem. You won't be aware of that processing, or maybe if it's some kind of pain associated, then it will reach your postcentral gyrus. Visceral motor comes out anteriorly. It will join information that is going to areas in that part of the body, maybe the body wall or some other location that is somatic, so going to skeletal muscle. They'll travel together to exit the spinal canal, but the information from the visceral motor will go into this sympathetic chain. It might rejoin the somatic neuron, but it may not. It may go up or down in the sympathetic chain and exit somewhere else. The white matter, not so lucky in differentiation between sensory and motor. The best that can happen is right here in this dorsal column that has two tracts in it. Here, this is mostly sensory information. Sensory information that is closest to the sulcus is coming from the furthest away. It's coming from your foot, it's coming from your leg. And then as you get more proximally, the neurons will be layered onto the foot and the leg. So you get thigh and hip next. And they color that purple for when you get to the hip. And then you get up to the trunk, and that will be layered onto the hip. And then you get to the arm, and that will be the most lateral. The arm and hand will be the most lateral that is in this posterior column. So posterior column, posterior funiculus. Funiculus is another name for tracts. You have a fasciculus that is in the center here, and a fasciculus that's in the lateral here. Those two on each side make up your dorsal column pathway. So sensory information, for the most part, not entirely, but for the most part in the back here, it's all sensory information. When you get to the lateral columns and the ventral columns, you've got a mixture of motor and sensory in those columns. So you're not so lucky. You've got to remember what type of sensation or what part of the motor system is in what part of the lateral and anterior columns. For the motor parts of the nervous system, gray matter, everything anterior is motor. So in the in the ventral horn, you have a similar parceling out of the particular cell bodies that are going to be involved in motor output. The difference is motor output goes down to the level it's going to exit, and then if you go to any lower parts of the cord, those cell bodies are no longer there. because they already are sending out information above the lower levels. So you're not going to see any representation down below. They're gone. They're finished. They've done their work. What you're going to see down below is lower extremity. Sensory information comes from your toe, to your foot, to your ankle, to your leg, to your hip, to your thigh, to your thigh, to your hip, to your trunk. Everything is layered, layered, layered on. It continues up until it reaches post-central gyrus. Going out, it's exiting at the level it needs to exit at. So motor information, you look at the front. What do you have here? You've got hand, forearm, arm, shoulder, trunk. Where's your leg? Where's your foot? Where's your big toe? It's not going to synapse with the lower motor neuron until it gets to the lower part of your spinal cord. So it's still running out here in your white matter. It's myelinated axon, still in the white matter. When you get to the level where the cell body is, then those pathways will come in here, synapse with the cell body of the lower motor neuron, information will leave. So nerves, these are all the peripheral nerves that you're going to not have to know all of the individual ones, but certain ones I have already boxed in red. A peripheral nerve is going to be consisting of myelinated axons with a lot of white around them. And then you look at these other areas that look darker, grayer. Those are unmyelinated axons. So myelinated, 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 myelinated axons. They're bundled together. Each of those axons will either have a Schwann cell that wraps multiple times to form myelin, or the axon will be supported by Schwann cells, this one time, one surface, one wrapping. I don't want to call it wrapping because it's just barely covering and supporting those axons. So endoneurium will be the Schwann cell and a little bit of delicate reticular tissue around the Schwann cell. And then you have a fascicle of axons, dendrites to me, and axons, the motor sensory bundled together. Those fascicles of afferent and efferent processes will be surrounded by perineurium, and then the fascicles will be bundled together by epineurium. So it's very similar to the muscle. Instead of a muscle fiber and its endomysium, you have a cell process, an axon, and its endoneurium. The fascicles are surrounded by endoneurium rather than perineurium rather than perimysium. And then the bundles of the fascicles will be surrounded by epineurium as opposed to epimysium. Okay, take a break and we'll talk about peripheral nerves and plexus.